Y bueno, ella es Malin Surkusk. Ella tiene 25 años de experiencia en microcalorimetría y varias técnicas microcalorimétricas y aplicaciones en estas. Su educación fue en bioquímica, fisicoquímica y biofísica. Tiene también una maestría en ciencias de la Universidad de Estocolmo y su doctorado lo realizó en la Universidad de Lund. La mayoría de sus estudios de doctorado los hizo en industria farmacéutica, donde trabajó con microcalorimetría isotérmica, DSC, también con ITC, así como otros métodos biofísicos. Después de concluir sus estudios, ella trabajó en Thermometric en Suecia, donde trabajó en el desarrollo de los instrumentos de microcalorimetría. Su trabajo incluía responsabilidades como gerente de producto, gestión de laboratorio de aplicaciones y marketing. En 2006, esta empresa en la que ella estaba trabajando, Thermometrics, fue adquirida por TA Instruments y ha estado laborando con nosotros desde entonces como especialista de aplicaciones en un inicio y ahora como gerente de producto en las líneas de microcalorimetría. Y pues agradecemos mucho a Malin que esté aquí con nosotros compartiendo su conocimiento. Esta sesión que vamos a tener el día de hoy va a ser videograbada. La grabación se las vamos a compartir al finalizar mediante un correo electrónico. Y las preguntas que puedan tener durante la charla las pueden redactar directo en el chat, o al final vamos a tener también un tiempo para atender sus preguntas y las van a poder hacer directamente a Malin abriendo sus micrófonos. Las preguntas las pueden hacer también en portugués, nos van a estar ayudando también ahí. Y al final, pues entonces habrá oportunidad de atender las dudas directamente con Malin, lo podrán hacer abriendo sus micrófonos. Mientras esté durando la presentación, les pedimos que para mejorar la calidad del audio de todos, tengamos los micrófonos en silencio. Y también quiero dar un agradecimiento especial al Colegio Nacional de Químicos Farmacéuticos Biólogos de México por esta colaboración que hicieron con nosotros. Específicamente quiero agradecer mucho a esta Elisa por invitarnos a colaborar con ellos y pues esperemos que esta sea la primera de muchas más. También quiero agradecer al equipo de TEA México que va a estar aquí apoyándonos, dando soporte si necesitan o tienen alguna duda. A Luis, al doctor Abel, a Sandra, gracias por estar aquí apoyando. Y también al equipo de TEA Brasil, porque tenemos a mucha gente de Brasil también aquí. Fred está por aquí, William también está por aquí, entonces William nos va a estar ayudando si tienen preguntas en portugués o necesitan alguna traducción al inglés, él nos va a ayudar. Y bueno, con esto quiero darle un poquito de tiempo a William para que les dé un breve resumen en portugués. A ver, William, adelante, gracias.
Muchas gracias, Damaris. Boa tarde a todos da América Latina, principalmente aos do Brasil que estão me ouvindo. Hoje a organização ela vai seguir, como vocês já perceberam, todo por conta do México. Solicitamos a todos que cerrem sus micrófonos, por favor.
Son 3 cursos, cada curso tiene su certificación. Si lo escribes por Sofía, cada uno va a tener su certificación.
Tenemos... Sim, agora sim. Essas coisas acontecem em reuniões online, né? Muitos em casa. E seguimos então, né? Se vocês tiverem dúvidas em português, por favor, podem digitar no chat, no final podemos abrir para discussão. Ou em espanhol ou em inglês, por favor, aqui a gente é poliglota. So now, Malin, now it's with you.
Okay, thank you. So I will start this presentation in pharmaceutical microcalorimetry. Let's see. So the outline of today's talk will be very, very briefly what is microcalorimetry. I will mention about stability and compatibility tests for pharmaceutical applications, both for small molecules, solid molecules, and for biopharmaceutical solutions. And then I will talk about how to determine small amounts of amorphic contents in a highly crystalline material. And the final examples will be on how to characterize polymorphism. So this is a 1-hour presentation, and we try to go over the most common applications during these presentations. There will be other type of applications as well. So after the presentation, if you think I have this thing I want to do, can I do it? And then we can have a discussion afterwards. There are many more things you can do within this field as well. I just try to give you an overview of the most common type of applications within pharmaceutical. So if we talk about calorimetry, we say that it's a universal technique because virtually all chemical and physical processes will result in either heat production or a heat consumption. So whatever goes on in the sample will generate some kind of heat flow. And this is what we measure with the heat flow calorimeter, and the heat flow calorimetry is the main focus of today's talk. And a heat flow is denoted the cubed T, so it's a rate of how much heat is produced. And this heat flow we are measuring is directly related to the heat production or consumption rate in the sample, which we denote T. I will talk mainly about heat production today during the talk, but it could be the other way around as well. Both the P, though the heat production and the heat flow is measured in watts, which is equal to joules per second or calories per second, depending on where in the world you are. And a microcalorimeter is simply a calorimeter that could measure that heat production in the microwatt range. So we usually, I hope you can see my what do you call it, mouse cursor. Maybe I should try to have a pointer instead. Oh. So we plot heat flow. In this case, it's normalized heat flow against time. And at any time, the heat flow is proportional to the heat production rate we are measuring. And the heat production rate of a sample is equal to the enthalpy of the reaction we are studying, the reaction rate, and a function of the concentration of your sample. For one process reaction, the heat production rate is related to the rate of the reaction. So if we would to do Arrhenius kinetics, we don't need to calculate the reaction rate. We can actually use the heat flow to plot to see the temperature dependence on the reaction rate. In this graph here, we have time of hours. So that's the shortest period you would do a microcalorimetric measurements, that's during some hours, but you can also measure for days, depending on what you want to measure or what information you want to have out of your system. So heat flow versus time relates to reaction rate. If we integrate that, we can plot heat versus time, and then we can see how well, the total heat produced that we have measured are related to the extent of the reaction. So we can see how far has the reaction come. So these are the two ways we typically plot the data. And from this you will get the enthalpy. Once the reaction rate or the reaction has stopped, you will have a flat line here, and you would then get how much heat that is produced for the total reaction that you're studying. Now calorimetry measure everything that goes on. So if you have several processes measured at the same happening in your sample, you will measure the total of all the reaction that goes on in your calorimeter. And now I will mainly talk about isothermal microcalorimetry, which means that we keep the temperature constant during the measurement. So we set the temperature to maybe 40 degrees and we measure what's going on at 40 degrees. We have a microcalorimeter system that we measure on. So we have a thermostat, that's we call the temp thermostat, where we will put different type of calorimeters into it. So we can equip from 1 up to 4 calorimeters to get either, depending on the needs, you equip it with different type of calorimeters. For more standardized measurement, like routine measurements, there is also 48, so you can measure 48 independent measurements simultaneously. You equip it with calorimeters, and then depending on how you're going to handle your sample, you either have closed ampules or you have what we call the microreaction system, where you can manipulate the sample during measurement. And then you will have some auxiliary. So you build your system. Most examples I will give today is with the closed ampule and also with what we call the RH perfusion ampule, where you can manipulate or change the relative humidity over the sample. So these are the main examples I will be giving today. So within pharmaceuticals or pharmaceutical science, there are different parts of the process of making a new drug. There could be calorimetry in all different areas, both during drug discovery and preclinical development. There we look at ligand binding, structure, function, relationship, also stability. That's used with isothermal titration calorimetry or differential scanning calorimetry. This will not be the focus of today. We will more focus on when you get to the pharmaceutical, when you have your leads to come into pharmaceutical development, pre-formulation, formulation, and analytical development, and also for production control, to look at purity, polymorphism, amorphicity, stability, compatibility. We can look at dissolution and hydroscopicity. So this, I say, is main thing. You can also see here we can do it both with DSC and isothermal microcalorimetry. And I will focus today on the isothermal calorimetry. DSC, I think more of you are familiar with to run DSC on pharmaceuticals. So this will be another approach to look at samples. So the calorimetric techniques, well, we will talk about IMC, isothermal microcalorimetry today. We will look at that for stability, compatibility, polymorphism, and amorphicity is the main topics of today. You can also do then differential scanning calorimetry. You have it, let's say, for the solid state drug stability. DSC is used in small pans and scanned. But for biopharmaceuticals, we also need new technology now for looking at stability in solutions. And then we have another type of DSC. I will mention that briefly during the talk later on, for looking at macromolecules in solution. There are different microcalorimetric techniques for looking at pharmaceutical applications. We talk about ampule calorimetry. This is where you just put your sample into a closed environment and you do no manipulation of your sample during the measurement. Just see if there is anything going on in your sample. So you can measure for stability, which is the pure substance, compatibility, if you mix your API with the excipients to see if they are compatible with each other. We can look at amorphicity and we can look at polymorphism. That could be done without manipulating the sample at all. Then there is possibility to use perfusion calorimetry, where you change either the gas composition, so expose it to nitrogen or air to see if there is oxidation or it's sensitive to the relative humidity that you can change during the measurement. It's also used to induce crystallization in amorphous samples to quantify how much amorphicity there are, and looking at stability of polymorphs. Solution calorimetry is not a heat flow calorimeter as these would be, the two first one. Here you will dissolve a solid sample and you measure heat increase or decrease while it's dissolved. This is used for assessing amorphicity. It's used for polymorphism and to look at interactions in solutions. So these are the main microcalorimetric techniques for solid state pharmaceuticals. So if we now quickly jump into the stability and compatibility and the importance of stability assessment, well, it's important to know that you have a stable drug when you send it out to the market. And you need to both test for the chemical and the physical instability, because if you have that, well, you can lose the potency of the drug. Degradation product may have bad effects. You can have undesired change in performance, and also the changes in physical appearance of samples if there is something happening. And of course, it's also a requirement by regulatory agencies. Microcalorimetry has the benefit because it could detect both chemical and physical changes. If you, like a standard method is HPLC or UPLC to look at stability, but then you need to dissolve your samples, and then you have no chance to look at the physical changes drug or formulation can have. But with microcalorimetry, you can detect both these kind of changes. And physical changes, they can be related to the polymorphism or the polymorphic behavior of your drug, to amorphicity and hydrate and solvent state. And all these needs to be tested on your actual solid formulation or solid drug. IMC is also very sensitive. In many cases, it's not needed to have accelerated conditions to get this compound to degrade faster. It's non-specific, which of course, if you see something, you may not know what's happening, but you would know that something is happening and you can do further analysis after that. And it's non-destructive, and it gives you real-time data. Of course, if you have a decomposition reaction going on, it will destruct because of this reaction, but you do not need to do anything else with your sample to measure it. And we have an example here of a drug measured. It's actually a tablet of a drug, a formulated tablet, measured at 50 degrees, 60 degrees, 70 degrees, and 80 degrees. At 50 degrees, you still see a flat line, and it's almost zero, but it is not zero. So it's actually an ongoing reaction that some sample, even if the reaction is very slow, we see there is a slow degradation process going on. If you see this and you don't know, oh, was this something, is it a real thing, then you can also increase the temperature and see if I increase it, yes, the signal goes up. 70, it goes up even more. At 80, we really start to consume the sample because it's degrading so fast. So we can follow these reactions. And again, we can do Arrhenius kinetics. We can take the reaction rate, or actually the heat flow, directly and plot that in Arrhenius to see if the same reaction we are studying at all these temperatures. Stability and compatibility tests. Well, I talk about stability and compatibility at once because compatibility is a type of stability, but it's where you combine it if one substance affects the other one to decrease the stability. Both stability and compatibility tests can be performed for screening, and you can do it to get quicker responses, to increase the temperature slightly and add a certain relative humidity. Very many stability and compatibility tests are done at 40 degrees with 75% relative humidity, adding a small microhydrostat. So that's in a closed ampule. You add a saturated salt solution to create a certain relative humidity. This stability test can be performed both qualitatively, like a yes or no, is it stable or is it not stable? Or you can rank different excipients or different formulations. And it can also be done quantitatively because you can also evaluate the kinetics of the process. So it could be a quick one, or it could be more involved process to evaluate the stability of a drug or a formulation. So you can do it on the API alone or in combination with excipients, and then you call it compatibility. It could be performed on the complete formulated product, and it could also be together with the packaging material. So you can put the drug together with the packaging material into the microcalorimeter, because heat will always flow out, and you will see if there is any interactions in between. You can also add relative humidity around the packaging material and see if it comes into your drug and affect the drug inside the package material. So this is an example, and those of you who have listened to calorimetry before, I guess this is a standard graph that we often show. This is an alpha tocopherol that's oxidizing, and it's been measured at 23 degrees, 30, 40, and 50 degrees. And we can see that the level increased by increasing temperature. So the reaction rate is increasing. This has also been put on the shelf and evaluated by HPLC, where you can calculate the rate constant. And if we have the rate constant from, or the reaction rate from the heat flow calorimeter, and we have it from the HPLC data, we can plot both these data in a Arrhenius relationship, and we can see that they fall on the same line. TAM data can go much lower in temperature because of the sensitivity for, it has sensitivity for relatively small changes in concentration. So you can usually detect that at lower temperature faster than you can do on ordinary shelf life studies. Another example is the stability of benzoyl peroxide, where it's been detected with UV-Vis, and microcalorimetric data, where you can calculate the reaction rate. We can see that we can go from 20 up to 45, but for the UV detection you need to go up to 55 degrees to be able to calculate the rate constant, or to be able to detect this. So we need to accelerate the conditions even more for doing other type of detections. We can see also that the microcalorimetric data, well, we can't see it here, but from the paper it says it was done within 16 hours, and that's a rate constant that's equivalent to a half-life of 22 years that you can measure at room temperature to see the decomposition or the breakdown of this substance. So for primary screen of stability is that you will, or the workflow, how you would do this test is to measure the heat flow over time of a known amount. You need to know the amount to be able to calculate how much is happening. And you will also decide that if I measure at 30 degrees or 40 degrees, I will say that it's not stable if it exceeds a certain number. So if you don't detect that heat flow, you will assume it's stable. If you have a measurable heat flow, then you assume it's unstable. So either you discard it as a new drug, or you will do more quantitative analysis to determine the rate constant, if I can shield the sample in some way to slow down the decomposition process. So you can do more work on it. For doing a compatibility test or with a binary mixture, the schedule would look like this, that you will measure the heat flow of the API alone. You will measure the heat flow of the excipient, and then you can calculate the theoretical heat flow if you combine this. If there is no excess reaction compared to the individual components, then you shouldn't have an incompatibility issue. Then you actually measure the heat flow, and if you compare the theoretical and the actual, and if there is no difference in data, you will assume they are compatible. If there is difference in the data, you will assume they are unstable or incompatible. Then you can either discard the combination, or you will do more analysis to understand what is this interaction. In our software that goes with the instrument, there are methods to do this very quickly, to calculate the theoretical. As long as you have the weights of the individual components, you can just input the weight and it will automatically make those calculations for you. So it could be like a screening, a fast screening. And then here's an example of a compatibility experiment with the TAM, where you have, in this case, we can also see how the relative humidity is changed with the saturated salt solution that put into the ampule. In this case, it's saturated sodium chloride solution, which will give a relative humidity of 75%. And this case is also measured at slightly elevated temperatures, like up to 50 degrees. We can see the drug and the excipient themselves gives very little heat, just below 0. And then if I mix them, I have an excess heat in this case, and then they would be not compatible. There is something reaction. There is a reaction between the components. So this was the first part. So to summarize that, you have the multi-channel capability with the different channels in either the 4-channel version of the instrument, where you can put 4 different calorimeters in, or you have the 48 version where you have 48 independent calorimeters to be able to have a more screening or high throughput. Microcalorimetry wise, 48 parallel measurements are high throughput. The high sensitivity gives the ability to detect interactions close or at or close to ambient temperature. If there is no interaction detected or the composition going on, samples are usable, or even if you detect an interaction, you can do further analysis of the samples afterwards. You have the ability to expose your sample to a relative humidity, and if you saw on the scaling, experiments may be long, but they are not as long as accelerated aging studies, where you put them on the shelf and analyze them. Now there is another type of stability that we can do in the microcalorimeter, and that's the stability of biopharmaceuticals. And biopharmaceuticals, like monoclonal antibodies, is the fastest growing field in biopharmaceutical industry today. They pose special challenges, but they usually require very high concentrations to be administered, like 100 milligrams per milliliter or more. And these high concentrations in the formulations will increase the risk of denaturation and aggregation, so you don't have a functional protein. And you also want to have methods to determine the best buffer excipient conditions to minimize this aggregation. And these biopharmaceutical formulations also should be stable for at least 1 year at these high concentrations. So there is a challenge in finding these methods, because traditional methods for the characterization of protein stability and aggregation often requires other physical or chemical conditions than the actual formulation sits in. So whatever test you do have very little predictive powers. So you will need to do shelf life studies real time to be able to determine stability of these molecules. And these methods that look at structural stability and aggregation will be DSC, DSF, ICD, so that's chemical denaturation, size exclusion chromatography, and light scattering to see how big molecules start when they start to aggregate. But all of these methods will require dilution of the formulations, and then you are back to this with long-term incubations prior to analysis. So there is a desire in the industry to find the methodology to be used on the actual formulation that could give you accurate stability data from the rate of the denaturation and aggregation. So I mentioned DSC, now we're talking microcalorimetry, so I would just mention the DSC as a typical method to look at this. Where you would then a DSC differential scanning calorimetry, so we change the temperature. So instead of hours here, we would have temperature on the x-axis, and we measure the heat flow, or in this case where we have normalized it by scan rate and how many molecules that's in the solution. We will measure the denaturation peak. And in many cases, when you screen for excipients and say that it's stable, you will have the TM, the midpoint of a formulation, to say the lower the TM, the less stable this formulation is. So that is a good way to screen for excipients. We can see that there will be an indicator of how stable a molecule is or the formulation is. So this is the same sample that's screened with different type of excipients, and we can screen them and look for TM. So it's a quick way of screening, but note also here that these are now denaturation, it's endothermic. After denaturation, some goes exothermic down, which means they start to aggregate. So even if this one let's see, we can see, this one looks less stable than the pink one. So the yellow one looks less stable than the pink one. We can see that the yellow one doesn't aggregate afterwards, while the pink one is aggregating afterwards, meaning that we are driving the equilibrium to aggregation. So you will both need to look for TM and also if there is aggregation occurring after the denaturation. Nevertheless, it's a quick way, but now if we would like to look at what happens if you isothermally store, I can also say that DSC in many cases need to be diluted below 10 milligram per milliliter to be able to put into the DSC, while we said that many of the formulation will be plus 100 milligram per milliliter, so we need to dilute them to be able to measure. So we change the conditions. The higher the concentration, the more prone they are to aggregate. We can look at two different mechanisms of protein denaturation or aggregation, that you can have a denaturation or a native molecule, it denatures and then it aggregates. If you run that in the DSC, you will see first the denaturation peak and then you have aggregation. So this is a DSC run. And then you can have a reaction where the aggregation occurs simultaneously as the denaturation. And then you will see this type of curve where it goes right down after. You actually don't know where the peak top is to be able to relate the TM in this case, because this reaction could be much bigger than the actual denaturation. So if we do, here's an example of lysozyme as a model system to test this. If we have the denaturation and then aggregation, what if we take a temperature just below the denaturation peak and put the sample in an isothermal microcalorimeter at a lower temperature and see what's going on? Is that possible? Let's see. And yes, it is possible to do that. So here they have put it at different temperature, 57, 58, and 59. We can see the denaturation is starting just slightly above 60 degrees. But even if you put them at temperatures below that, we can follow now we have an endotherm and an exotherm. So endotherm is the denaturation, exotherm is the aggregation. So we can follow that process. Now this has been measured for 6 days. This is how the formulation looks when it's put into the isothermal microcalorimeter. When we bring it out, everything has aggregated. So we can follow the process calorimetrically. This exotherm will be bigger at higher temperatures. So the process is faster. And then we can fit those, we can make models, because the heat flow, as I said, is proportional to the reaction rate. So we can have the reaction rate, or we can make a fit where we evaluate the reaction rate first for the aggregation and then for the old folding. So that could be fitted. And if we have fitted this data at a different temperature, we can calculate a reaction rate for unfolding and reaction rate for aggregation. If this now is plotted again in an Arrhenius relationship, where we take the reaction rate versus the inverse of the temperature, we can say that this will correspond to a reaction rate that it will be aggregated in 350 days. So this is a way of doing it. We have a real example as well with a monoclonal antibody, where they have run, now they have screen for different buffers. We can see that all buffers in the DSC, they will aggregate immediately after unfolding. Then they could put it in the microcalorimeter at temperatures that then slightly below when the peaks start, and we can see the different buffers. With the different buffers, this monoclonal antibody will be different. Now we can see also that we don't have the endo-exothermic, because this is a reaction that happens simultaneously. So we actually see more of the endothermic reaction here compared to the exothermic aggregation here. The more heat involved in the denaturation. Now maybe this is a little confusing, because in all other graphs I've showed you in isothermal microcalorimetry, exothermic is upwards, but from this particular paper they have published endothermic data upwards instead of downwards, so these are changed twice. This has also been stored on the shelf. You can see the time here is 8 days. This has been stored on the shelf, and then size exclusion chromatography has been made after 12 weeks. And we can see that if we increase the graph here, the aggregate peak is increasing, has increased, or there is aggregate peak with the different buffers. These different buffers will correspond the colors with the stability measured in the isothermal microcalorimeter. So again, data could be fitted with this non-least square fitting, and we can get the reaction rates by building these models of unfolding and aggregation. We can see different buffers depending on concentration. The higher the concentration, the faster the denaturation goes, or the aggregation in this case. And they could then rank the buffers correspondingly by the reaction rate. If we know the reaction rate for how quickly it aggregates, we can also calculate how long time it takes for the protein to aggregate at these measured temperatures for the different type of buffers. We can see there is a difference between the buffer compositions. This can then be compared with the size exclusion chromatography, where you put the percentage of aggregate versus the rate, and we can see we have a correlation and the same ranking of the different buffers in this graph. So we can also conclude we have a correlation, but the size exclusion has been done in 10 weeks and all the TAM test has been made in 10 days. So we can, for ranking this and see better or less good buffer, it's much quicker to be able to do this with, I say TAM, this is the isothermal microcalorimeter that's used for this. So that's the name of our calorimeter. So to conclude this, that microcalorimetry has the possibility to detect the denaturation and aggregation kinetics at temperatures below the TM. You can get stability assessments in a few days, compares for weeks for other storage tests. It could detect the heat signal in small volume. In these cases, we have seen they've done 1 milliliter, less than 1 milliliter, and at high protein concentrations. But we also saw that some of the samples were down to 25 milligram per milliliter, so you can still also measure at lowers, but there is no need to dilute. You can get a higher sample throughput, so you can screen a lot of different buffers and excipient conditions simultaneously. And they are quicker than today's conventional methods to identify the formulations to have best long-term stability. And in addition to that, as we mentioned, these other type of methods, they measure the existence of aggregates after storage, while we are measuring the rate with the microcalorimeter. So we have the potential to predict the time of storage. We don't need to wait till the process is finished. We can predict it during the process. So that was on stability and compatibility on both solid state pharmaceutical and biopharmaceutical formulation, that liquid formulations. Then there is another one. We'll come back to the solid state application where we can look at amorphicity. Well, amorphous drug substance can be produced intentionally because the amorphous form will have an increased bioavailability. But then, of course, you need to demonstrate the stability of that formulation. What we also can do with microcalorimetry is to detect small content of amorphous material in a highly crystalline material. That could be created during the processing, but since these process-induced amorphous region are on the surface, they can have a great impact on the final product. So it's very important to have control over this. And also, if you have this amorphicity, it will affect the properties of the drugs. So there are different ways of detect this low level of amorphous content, because the imperfection, the amorphicity, will increase the energy of the crystal that you see. And so we can measure this calorimetrically, either by the heat of crystallization. We can induce crystallization by humidity, so we lower the glass transition and induce a crystallization of small amounts of amorphous content in our formulation. It's possible, I put this in a little gray here, that we can measure heat of absorption by measuring water or solvent absorption without inducing crystallization, because there will be different absorption behaviors. I will not talk about that today, but that's a method we see that more and more is used. And then we can do it by heat of a solution, measuring the enthalpy of when I dissolve the substance. These two methods, I will briefly talk about them. There is a USP method that describes both these methods, also how to quantify or detect this. The simplest way and the most, we'll say, high throughput method is, again, using a closed ampule and put a small microhydrostat in. So when you put a highly crystalline material here, you will not, if there is no amorphous content, you will not get a peak here. If you have small amounts of amorphous content where you induce the crystallization, you will put it into the calorimeter. You will measure first absorption of humidity or another vapor from something that will be absorbed to your molecule or your drug. And then once you have absorbed everything to lower the glass transition, you will have a peak of crystallization. If you get this peak, you know you have small amounts of amorphicity in your sample. But you can also integrate and quantify how much amorphous content do I have in the sample. You can see on the time scale here that we get the peak very quickly. This is more or less a 2-hour measurement. This time will depend on the vapor activity, the higher water activity or the vapor activity. The faster the peak will get, because it will faster absorb all the water it needs to induce the crystallization. The temperature, the higher the temperature, you will get the peak. Sample size and the presence of crystals or seeds. Can you just show an example how the vapor activity is quicker for one, or if you have 100% relative humidity, the peaks come quicker than if you have a lower humidity. So depending on if in this case you have pure water or you have sodium bromide, that will create this relative humidity. Sample sizes here is about 30 milligrams, so you don't need to use very much sample, and the sample temperature was 25 degrees. So you can see how you can set the time to the peak depending on choosing the vapor activity. The bigger the sample, it also takes longer time for it to absorb all. So you play both with humidity and sample size in this case. Then we can also, this is a fixed relative humidity, what we just saw in the previous slide, but you can also change the relative humidity during the measurement. Then we need to use the perfusion ampule, the RH perfusion ampule, where we can change the relative humidity. So this is an open system. It's one part of the microreaction system where we can manipulate our sample during the measurement. Here we will have two gas inlets. One will go directly down into, this is the ampule where you have your sample that sits inside the calorimeter. Top part is outside the calorimeter. So one part goes directly in, one gas line directly into the sample, and one line goes through the humidifying chambers. This chamber sits in the thermostat, so they have the liquid. Now we talk about water and humidity, it could be other solvents as well, but it will have the water, so we will humidify the air at the measuring temperature before it goes into the sample chamber. So we can play with how much gas that goes through the dry line to the wet line, and we can create the relative humidity over the sample to determine where I want to measure. So we have possibility to run at the constant defined RH. We can stepwise change the RH, or we can ramp the RH over the sample. We have an example here where we ramp it, so from 10% relative humidity up to 80%, where we have first absorption of humidity, then we have the crystallization of the amorphous part. After it's crystalline, it doesn't want to hold water anymore, so you can see this endothermic peak is where it expels the liquid. At higher relative humidity, you have a transformation of the, in this case, the lactose. So we can follow, this is a little more difficult to quantify, but you can detect that you have amorphous parts in your material. A very sensitive method to look at the amorphous contents is again to use the RH perfusion ampule, but then you step between certain relative humidities. You will now equilibrate the whole thing at 30% relative humidity, and then you step up to 40%. So you get the peak absorption and maybe crystallization if you have a amorphous part. Then we can step down back to 30, we get desorption, and then we can step up again just to verify. If I now move this peak here, where I go from 30 to 40 to here, we can see that they are not completely overlapping. So between 89 for the first peak to 69, that difference is related to the crystallization of the amorphous material. By doing standard curves for that, we can conclude in this sample, it was 0.3% amorphous content in this sample. So it's a very sensitive method to look for amorphicity. The other method we have is solution calorimetry, where in the solution calorimeter, we put our solid sample in a small ampule, and we crush this ampule within a bulk liquid, and we measure the heat of this solution. The solution process is typically endothermic for a crystalline material, and it will be exothermic for an amorphous material. So we measure temperature change, and that could relate to how much heat is either produced or consumed while it's dissolving the sample. And then again, you will have to do standard curves, and the sensitivity of this technique will depend on how big difference it is between the completely crystalline material compared to the completely amorphous material. So if there is a very little difference, you don't have a high sensitivity. If there is a big difference, you can get quite a good resolution of the data. So to summarize these different methods I mentioned here, is the microhydrostat method, the CRHP method, where we stepwise change the relative humidity, and the solution calorimetry method. We can see that the stepwise relative humidity change is by far the most sensitive, but we can, with a good setup. You can get very low in sensitivity for amorphous contents also with the microhydrostat method. Note that these two methods, they will tell you something about the surface that's accessible for the humidity on your particles, amorphous part. But of course, that's often what's happened when you process. It's the surface that you change. The SoCal method will dissolve the whole particle, so you will see if there is amorphous part also within the particles in this. So you can get the time scales, how much sample you need for the different measurements, and how many simultaneously. So the microhydrostat, of course, is a more throughput method as well. And both these two are USP methods as described in that. And we will see that this RHP is more for R&D to evaluate what's going on and when it's going on. Okay, so that was amorphicity, and now we come to polymorphism, and of course that's also extremely important to have a control over, because different polymorphs can have very different properties, and it can affect everything from bioavailability, stability, compactability, and also safety of the molecules. And how would you use microcalorimetry to characterize this? Well, we can use microcalorimetry to determine the relative stability of polymorphic pairs. If we have solubility data from another test, we can get transition temperatures. It could also tell us if the powder is in a metastable state, that it's transforming over time, and we can also see which factors that could stabilize or destabilize the system. So it could be used as a process monitor to watch what's going on with the molecules or with the samples. So just start with the relative stability of a polymorphic pair. So different forms would have different heat of dissolution. So we can determine the relative stability of a polymorphic pair by calculate the heat of transfer between the different polymorphic forms. So, well, you can do this with a DSC. You can get the transformation by increasing the temperature and get the transformation from one polymorph to another. We can also do it then by dissolution calorimetry. As I say, we put it in an ampule, and we will crush the ampule in a bulk liquid, and we measure the heat of dissolution. So we can measure the heat of dissolution of polymorph A. We can measure the heat of dissolution of polymorph B, and by Hess law, we can then subtract these from each other to get the transformation enthalpy. So we can take different paths, and we get the enthalpy of transformation between them. And then, depending on if it's an exothermic and endothermic reaction, we can say that A is more stable than B, or B is likely more stable than A. Now I say likely because now we are only looking at the enthalpy, but as you're aware of, the Gibbs free energy will determine if the Gibbs free energy is less or more than 0, would actually determine the direction of the reaction. However, well, say in the delta G you have enthalpy minus T delta S. But the entropy will be the same if your substance come out in solution. So the entropy effect will be cancelled out here, so we can look at the enthalpy effect because it comes to the same final condition. And if you have the solubility of the two forms, you can also calculate the transition temperature. At what temperature will this drug transform from A to B or B to A, depending on which is more stable than the other. This is described more in these two papers that came out approximately simultaneously, that describes two different approaches to calculate the transition temperature. Then we can also study the polymorphic stability to see if polymorph A goes to polymorph B. We can follow that over time by just putting them into an ampule in the calorimeter and follow the process. The dry, if we just put the powder in, it could be quite slow, but it can also be done via solvent-mediated polymer phase, where we can we make a slurry, and the least stable polymorph typically will be dissolved, and then it will crystallize into B. When this has disappeared from the solution, we dissolve more of A that crystallize into B. By doing this with a slurry, we can force A to go to B, and we can make it happen faster. So we can follow this process over time, going from one polymorph to another. But in this case, you can also see in the graph here, it's been measured for quite some time. But this is slow, but you can make it run much quicker. And this is an example that was from one pharmaceutical company, where they have different batches or change processes, where we can see that depending on what sample they put in from which batch, we can see one will transform very quickly from one polymorph to another, while depending on how it's been treated or what's put into the process, you can actually make the process go slower over time. So this is a way also, as I said, to start with, to have it as a process monitor. Can I put something in to make this process so I can stabilize my metastable polymorph to be stable over time? Enthalpy of all those processes are equal if you integrate the areas. So it's the same process, it's just the kinetics of the process that is manipulated here. So to conclude, the benefits of using microcalorimetry to look at pharmaceuticals is it's universal. If anything happens in the sample, it's likely to be detected. It's non-specific, so everything that goes on in the sample will be detected. You may not know what it is, but you know something is going on. Typically, you don't need to do sample preparation. Of course, you can do sample preparation to get more information out of it, but principally, you just put it into one of these ampules, put it into the calorimeter and watch what's going on. It's not depending on the physical state of the sample, as long as you can fit it into our ampule. These are 4 milliliter ampules. I said you can also use it with packaging materials. We do have 20 milliliter sizes, so you can get bigger type of samples in packaging material, maybe into the calorimeter. It's non-destructive. You can do further analysis afterwards. It's direct and continuous, so you get real-time data. It's highly sensitive. You can do measurements or detect instability, for example, at or close to ambient. You don't always need to do accelerated conditions, and you can have high throughput with up to 48 simultaneous and independent measurements. So now I have given a very quick overview of the different methods here. If you want to know more, on our homepage, we have some webinars to look at. Many of the slides here come from a webinar that we have on our homepage where I did on pharmaceutical science. Some of the parts where we look at the biopharmaceuticals, there is also a webinar for that. There is another just recently looking at details on the drug development using microcalorimetry with my colleague in the US. There are some introduction on how to do microcalorimetry, and then there are some what we call tech tip videos, how to prepare different types of molecules. So with that, I will end my talk. So if there is any questions, I'm happy to do that.
Thank you, Malin. So everyone that is in the talk, If you have any questions, you can just open your microphone and ask directly to Malin.
Malin, I have a question for you. In your last slide, you mentioned about monoclonal antibodies.
Yeah.
Is that related to the antibodies for like COVID situations now?
Well, yes. Well, for COVID, if they look for antibodies there too, that could also be an application where you actually look at if you are creating drugs or antibodies for that. Yes, you can evaluate both stability and formulation of those drugs to be for that application. Yes.
Okay, thank you.
We are also can also mention that in many cases, if you have a human antibody or another drug that you want to make a similar, that you want to produce recombinant or produce in other ways, DSC is also a tool to be able to compare, like a fingerprinting, because a DSC trace of an antibody is quite unique, like a fingerprinting of that. So if you're going to produce new antibodies that should be equal to let's say the well, I can't say real, I don't find the word, but the natural antibody, then you want to produce it. If you want to compare them, you can do that with DSC also to get sort of a fingerprinting, that you get the same kind of profile of it as well. I'm not showing any of those kind of data in this presentation, but it will be shown in that if we go back to.
So in that kind of development to try to mimic a natural antibody, that's when the TAM48 becomes a very powerful tool because you run multiple conditions to arrive to a particular expected result?
Yeah, well, the TAM4 or the TAM48 will look more at stability, but I'm thinking about the DSC that I mentioned a little briefly in the beginning. So microcalorimetry DSC, where you scan over temperature, that's used to fingerprint to see structural functionality that's in the system. But then you need to do it in dilution. With the TAM48, you can look at stability, and you can have a screening of stability on the actual formulation.
Very interesting.
Malin, I have a question also. Do you have of any pharmaceutical company that is using the TAM4 controlling the temperature and also the humidity to imitate the accelerated stability tests that are required to be able to sell a medicine?
Sorry, what the accelerated?
Yeah, in the pharmaceutical companies to sell these products, we need to make this specific accelerated stability test, where you need to control the temperature and also the humidity, but they commonly last like 6 months to get that information. And in the TAM4, you could obtain this information in a really short time. Do you know if any company is using the TAM4 for this kind of experiments?
Yeah, well, it is used in a lot of different big pharma companies. They are using it for this kind of application. However, it's not, I think when they file for a new drug, they still need to put in this long-term stability test. It's not it will not be approved, but it's a supplementary information, then you put it in. But what it's also used for is that you can screen to start with to know that I don't need to put this substance on the shelf. I don't need to put that combination on the shelf. So you can minimize the number of failures after 6 months if you have already tested it in the TAM.
Yeah, that's the big advantage I see in this technique, because you will know that information before. So you won't be wasting your time waiting for the test to be ended, the accelerated stability test to be ended. So you will have this information before. And I can see the powerful tool we have to have this, this information at a really good time. So I hope in some time near in the future, we can use this type of test that takes so much less time. And I hope the FDA and in Mexico, the COFEPRIS will approve this kind of results because this will make a development so much short in time and also cheaper. So I hope we can get enough data to prove that the kind of information we can have from the TAM4 could be, I don't know, it could be the same that we are going to obtain in an accelerated stability test. I hope that happen sometime.
Yes,
Yeah,
I
that would
agree.
be awesome.
Yeah.
Does anyone have a question here in the audience?
William, do you have any questions from the Brazilian community?
Actually, I was looking at the meeting chat and there is none.
Okay.
Or you can ask them in Portuguese if they...
Yes. No, but we have a little bit more to talk about it yet. So I think we are over, right?
Okay, yeah, I think we are.
So I'm doing an announcement for the Brazilian guys. Hold on. Pessoal, vocês que entraram agora, eu percebi que a gente cometeu um pequeno engano na publicação dos horários. Houve uma confusão, ok? Mas esse arquivo ele vai ficar disponível depois no nosso site, aqueles que quiserem assistir depois. Essa apresentação vai estar disponível lá no Take Talks. E a gente também vai deixar disponível a apresentação em PDF, como a gente sempre faz, ok? Não se preocupem, a gente pede desculpas pelo mal entendido, realmente essa diferença de horário pegou a gente pelo pé, né? Essa história de ter ou não horário de verão gera uma confusão para a gente. Mas fiquem tranquilos, a gente vai resolver isso, tá bom? E para a próxima semana a gente vai melhorar essa discussão para ela não acontecer mais, ok? Se vocês tiverem alguma pergunta, aqueles que entraram ainda conseguiram assistir, por favor, podem fazer pelo microfone ou no chat, pode ser em inglês, pode ser em português, e a gente passa a pergunta para a Malin, ok? Obrigado.
What happened, Malin, is that as we saw close to the end of your presentation, a significant number of participants from Brazil began to join the presentation, and it was due to the scheduling that sometimes the tools that we have do not adapt the translation to the times, to the other time zones.
Okay.
So some of the participants that joined were joining for a seminar that was supposed to be starting a few minutes ago. But William just told them that not to worry, your presentation has been recorded and they will receive a copy of the whole presentation, not just the last 2 slides that were quite interesting. So,
So Malin, we do have a question here. A colleague of us is asking if there are some of those equipments already in use for approbation in pharmaceuticals industries or researchers. Are they in use, right? They are already being used.